Hello friends, welcome back to my new YouTube channel Bio Content. Friends, in today's video we are going to discuss about the great scientist that is father of genetics Gregor Johann Mendel and his laws. What he contributed in the field of genetics that we are going to discuss. Friends, became the father of genetics, but how he became the father of genetics? You have to look into his contribution in details. That today we are going to cover his life, how he completed his education, where he served as a teacher, what was his family background, how he became priest in Australian monk. That thing we are going to see. Gregor John Mendel was born on July 22, 1822, near Brunn. Now it comes in Czechoslovakia. He was born in the poor family and his father was farmer. Mendel had a definite interest in living objects in his early life, and his father had a great love for plants, and his uh, uh, these things undoubtedly influenced the Mendel. As Mendel grew older, he became interested in plant hybridization. We always say the atmosphere affects us. The same thing happened with the Gregor Johann Mendel because he grew with his father, and his father had the great interest in plants. Because of his father's great interest, he could develop his interest in plants. With the help of his experiments, which are known to the world on Garden P, he was able to formulate two laws known as Mendel's laws of inheritance or Mendelian inheritance, which explain the manner of inheritance of characters, how the characters are transferred from parents to offspring. His early life was not so simple. He was unable to continue his study. As I said, he was from the poor family. Due to poverty, he joined the St. Augustinian monastery at Brunn in 1843, and he became there as a priest. After that, he was sent to the University of Vienna, where he studied the physics, maths, and philosophy. And that study was completed because of her his sisters. Sisters got given them the token amount for completing the education. Then he returned back to the Brunn in 1854, where he was appointed as a substitute science teacher. And his performance as a science teacher was outstanding. In addition, he worked as a priest in the local church. He lived in a house located within the premises of the church where he could completed his experiments on plant hybridization. After spending some time as a science teacher, he started working on the garden pea. He collected the several commercially available peas in the entire Europe, and he started his experimentation in 1857. He conducted all his experiments within the kitchen garden of his house with the help of his own resources. But his experiment or his findings uh, was kept for some time unknown to the world. After 7 years, he presented his findings in the Natural History Society of Brunn in 1865. That paper entitled Experiments in Plant Hybridization. Mendel not only worked on the Pisum sativum, he studied the French bean and some other plants, honeybee, and climatology. It was a tragedy with the Gregor Johann Mendel. Why I'm saying tragedy? Because 16 years after his demise in year 1900 only, the three scientists working independently, the Hugo de Vries, he was from the Holland, Karl Korens from the Germany, and Erich von Disselmarck in Austria. These three scientists started working independently on the same topic which Mendel worked. Although the basic principles of genetics were enunciated in 1865 by the Gregor Johann Mendel itself, but the new baby born was kept in an incubator and forgotten for the almost next 35 years. These three scientists made the science of genetics available for all of us. Why Mendel selected P as an experimental material? Because some features are there, some characters are there with the P's. Because of those characters, Mendel selected a P as an experimental material. In the pea, many varieties were available commercially. The flower size of the pea was relatively large. Because of it, the Mendel could manually emasculate the flowers of peas very easily. The flower structure of pea ensures the cell pollination, and in another and simple word, we can say the nature of pollination in a pea is a cell pollination. That's why there is no chances of cross contamination in that varieties. And the fourth uh, thing is the duration of pea. It can complete a pea can complete its entire life cycle within a season. That's why many generations can be grown in a year. Pea seeds are large and present no problem in germination. Because of these five reasons, Mendel selected the pea as an experimental material. Why Mendel got success? Some reasons are there. Mendel studied the inheritance of only one pair of contrasting characters at a time. One pair of contrasting characters means, for example, tall and dark. Tall is one character, height is a character having two contrasting form. One is tall and another one is dark. He selected P varieties that had clear differences in one or more character. For example, round versus a wrinkled seal. These are the clear differences in between the shape of the seal. Mendel classified all the plants of a population on the basis of contrasting characters under study and kept an accurate record of the number of plants in each category. Means while experimentation, he maintained the pedigree of all the generations along with the parental characters. His knowledge of math was a definite asset on interpretation of his findings on the basis of laws of probabilities. You know, the knowledge of statistics is very much important to interpret your findings. And that was the great asset with the Gregor John Mendel. And another reason is he was able to accept the ratio ranging from 2 is 2.82 is to 1 to 3.15 is to 1 as an overall estimation of 3 is to 1 ratio. 3 is to 1 is the phenotypic F2 ratio when he made a cross in between contrasting characters. Mendel was lucky. Why we are saying the Mendel was lucky? Because the 7 characters selected by Mendel showed the qualitative inheritance. Qualitative inheritance means that has the discontinuous variation from one generation to another. Each character is governed by a single major gene with complete dominance. Friends, there are two types of characters, qualitative and quantitative. Quantitative characters are governed by many genes and has the continuous variation. But in case of qualitative character, it is governed by one major gene and has no environmental effect on their expression. Of the 7 characters, the gene for 2 characters were located in one chromosome, while the three others were in another chromosome. These are the reasons why Mendel was lucky and he could formulate the laws of inheritance. Mendel selected the 7 contrasting pair of characters. What were those? And it is already studied a dominant and recessive trait. Dominant trait means the trait which can be expressed in F1, and the recessive trait means the trait which is suppressed by the dominant one. That's why could not able to express its own character. These are the 7 characters Mendel studied. That is plant height, as I said, tall versus dwarf, seed shape, round versus wrinkled, seed coat, yellow versus green, seed coat color, green versus white, pod shape, inflated versus constricted, pod color, green versus yellow, flower position, axial versus terminal. Above these 7 traits are dominant in nature over these below traits. Friends, what is meant by monohybrid cross? Mono means single. Hybrid means it's a progeny. A cross between two parents differing for a single character. Mendel selected the two parents. One was a round seeded variety and another was a wrinkled seeded variety. He made the cross in between these two, round versus wrinkled. The genotype of round was capital R, and the genotype of wrinkled seeded variety is small r. When he made a cross in between a round and a wrinkled genotypes, he could able to get heterozygous genotype. But the heterozygous genotype is having a round seed shape. Why it's round? As I said, capital R is there, which is dominant in nature, and it is having the round character. That's why in F1 only the round seed shape is expressed. After F1 generation, that is first filial generation, we can have the F2 generation, that is second filial generation. How the F2 generation is produced? By selfing the F1 series or by making the cross of F1 into F1. You can able to find out the F2 progenies. These were the genotypes of both F1s, heterozygous in nature. After forming these 4 genotypes, having 3 round-seeded plants, he could able to produce, and one was wrinkled-seeded. Genotypic ratio of F2 is 1 is to 2 is to 1, 1 capital RR type, 2 heterozygous, that is capital R small r, and 1 small r small r. These are the 4 genotypes. What will be the phenotypic ratio of these 4 genotypes? 3 round and 1 are wrinkled. What is a cross between parents differing for two contrasting characters? Here, two contrasting characters are considered: yellow and round seeded. Yellow color of the seeds and round shape of the seed versus green and wrinkled seed shape. The F1 was capital Y, capital R, small r. As the capital Y is dominant and capital R is dominant, the F1 will be always yellow and round seeded in nature. As we have produced the F2, here by producing the F2s, you can able to find out the F2 phenotypic ratio likewise. 9 genotypes will be of yellow and round seeded, 3 will be of green and round seeded, 3 will be of wrinkled and yellow seeded, and 1 will be of green and wrinkled seeded. These are the 16 genotypes produced or developed when considering the two contrasting characters. Out of these 16, likewise, the phenotypic expression will be there. What is dominant character? As we have discussed, the characters which appear and express in F1 is known as a dominant character. And another character is recessive. Those parental characters which are entirely disappear or suppressed in F1. Some terminologies must be understood by all the science students. What is gene? The term gene was coined by Johnson in the year 1909, and it is the hypothetical unit of inheritance. But friends, gene cannot be seen with your naked eyes or cannot be seen by using the electron microscope. Gene is nothing but the base pairs. It may be in the form of capital T, capital T, or small t. This is just for your explanation. What is allele? Allele is alternative form of a gene. Hmm, It may be capital T or small t. What is homozygote? An individual derived from the union of gametes of similar genetic constitution. Means the genotype, if it is homozygous, it may have such type of genotype. That is capital T, capital R, or small r. It must be homozygous in nature. What is heterozyte? An individual derived from the union of gametes of dissimilar genetic constitution may be either capital R, small r, or capital D, small d, called the heterozygotes. What is genotype? Genotype is nothing but the entire genetic constitution of an organism in respect of particular trait is called the genotype. Genotype may be of like this capital T, small t, small t. Any type of genotype can be seen. What is phenotype then? Phenotype is nothing but the external appearance of an organism is known as a phenotype. Friends, Mendel could able to formulate the three law of dominance, law of segregation, and law of independent assortment. Now we'll see one by one. What is law of segregation? When a pair of contrasting characters are brought together in a hybrid, the factor responsible for the characters do not blend or contaminate each other. That is the most important thing in the hybrid. But when gametes are formed, they segregate and pass into different gametes in a definite proportion. This law of segregation can be explained with the help of monohybrid cross. And this also explains the purity of gametes. You can see here when the Mendel crossed the two round and wrinkled seeded varieties of pea. The round seeded variety was having the genotype capital R, and wrinkled seeded genotype was having smaller. When he made cross in between these two, he could able to develop the heterozygous genotype having the round seed shape. By producing the F2 progenies, he could able to produce the four progenies. Out of these four, three were round seeded and one was wrinkled seeded. The phenotypic ratio will be in this manner 3 is to 1, 3 round seeded and 1 wrinkled seeded. Second is law of independent assortment. Law allele of different genes assort independently of one another during gamete formation. Each and every gamete will inherit or pass from one generation to another independently of one another during gamete formation, as we have explained this while explaining the dihybrid ratio. The same thing is applicable here. Now, the after Mendelian inheritance, some non-Mendelian inheritances are also there. Means, in simple word, we can say exceptions to Mendel's laws. It is deviation from the Mendelian inheritance. How we can say this is the exceptions to Mendelian inheritance? Because the phenotypic ratio which is expected in Mendelian inheritance in case of single cross 3 is to 1, and in case of dihybrid 9 is to 3 is to 3 is to 1. But in case of such type of some non-Mendelian inheritance, you may not form 3 is to 1 and 9 is to 3 is to 3 is to 1 phenotypic F2 ratios. That's why we are saying it's a deviation or non-Mendelian inheritance. The example is incomplete dominance. When Mendel, this example uh, is given from the Mirabilis Jalapa. When the scientist could cross in between the red flowered and white flowered genotype, he could able to find the different F1, neither similar to parent nor the blending of the both parents. It's a something different. This is intermediate uh, character of this parental character. Red versus white. The F1 was pink in nature. The genotypic and phenotypic ratio will remain same. As I have just explained, the phenotypic ratio is deviated from the Mendelian inheritance. In case of Mendelian inheritance, 3 is to 1 ratio is expected, but here 1 is to 2 is to 1 ratio is being showed in case of incomplete dominance. The second exception is codominance, where both the parental alleles will express fully in determining the phenotype of the F1. That's why the roan color of the F1 is seen. When the pure red and white uh, breed of cow were crossed, the F1 was roan in color because of fully expression of the alleles of both the parents in F1. That's why the phenotypic and genotypic ratio will always will be same. That is 1 is to 2 is to 1. This is the deviation from Mendelian inheritance. Again, the ABO blood group is one of the example uh, exceptions to Mendelian inheritance. Again, many examples are there. Maternal inheritance is there. Multiple allele is there, where you can see the variation in Mendelian inheritance. Friends, this is regarding the Mendelian inheritance, Gregor Johann Mendel and his laws. I hope you understood the topic, but whether you understood or not, that must be checked by yourself. So, Here I have given you some multiple choice questions to check your uh, knowledge, whether you understood or not. Who is the father of genetics? You might be knowing. How many laws of inheritance did Mendel propose? Which of the following is not one of Mendel's laws? Friends, attempt all these questions and check your knowledge. Which of the following best explains the law of independent assortment? Mendel conducted his experiments on which plant? A homozygous dominant pea plant is crossed with homozygous recessive plant. What is the genotype of the F1 generation? All the answers must be answered in comment section. What is the phenotypic ratio in the F2 generation of a monohybrid cross? The dihybrid cross ratio of 9 is to 3 is to 3 is to 1 represent which of Mendel's laws? In Mendel's pea plant experiment, which trait was dominant? Find the answers and try to mention in the section. Thanks for watching.